Hi, everybody. My name is John Long. I'm a faculty member in the Department of Pathology and the Stanford ChemH Institute. I'm delighted by the opportunity to present here today and for the support from the Knight Initiative. Our project is about capturing the molecules of exercise for brain health and brain resilience. Our team is composed of myself, Tom Montine, chair of the Department of Pathology, and Stephen Bannock, a faculty member in the Department of Chemistry and also my colleague in ChemH. We draw from the fields of metabolism, neuropathology, and chemistry, and we're extremely excited to take this multidisciplinary approach and with support from the Knight Initiative to tackle the problem of brain resilience. We all know that exercise is good for you. In fact, increasing physical activity has been shown to reduce cognitive decline with aging. From multiple clinical studies, that effect can be dramatic, up to 30% reduction in Alzheimer's disease. Conversely, physical inactivity is a major and modifiable risk factor for the development of neurodegenerative disorders such as Alzheimer's disease. Despite that robust clinical literature, the molecules and mechanisms that link exercise to brain health still remain relatively mysterious. Our team recently discovered a previously orphaned exercise-inducible metabolite called Lac-Phe, the structure of which is shown on the right here. What's special about Lac-Phe is that it's a conserved exercise-inducible metabolite that is a hallmark of exercise, regardless of exercise modality or mammalian species. We looked at humans after resistance exercise, after aerobic exercise, after sprint exercise. We looked at animals that move, including, for example, racehorses, sled dogs, mice, and rats. No matter what you look at, a hallmark after exercise is the induction of this Lac-Phe metabolite. But Lac-Phe is not just a hallmark of exercise, it is in fact an effector of physical activity. We were able to show in subsequent experiments that Lac-Phe actually acts on the brain, where one of its actions is to suppress feeding behaviors and food intake. We think that Lac-Phe might be why, after a hard workout, you don't feel hungry. But what's more important for the purposes of our proposal today is that we believe that Lac-Phe provides a clean chemical and molecular handle to understand the effects of exercise on the brain. Our team is interested in answering the question of whether Lac-Phe might mediate other positive effects of exercise on brain health beyond appetite regulation alone. For example, we're interested in cognition, learning and memory, and many other behaviors and phenotypes have typically been shown to be affected by physical activity. The reason that we think this might be possible is because we have genetically engineered mice to not be able to produce Lac-Phe after exercise. Beyond dysregulation of feeding behaviors, the other thing that these animals show is reduced learning and memory in two different assays, the novel object recognition assay and fear conditioning. Learning and memory are two cognitive domains most impacted during Alzheimer's disease. And so we believe that based on this preliminary data, we might be able to show a more broad role for Lac-Phe in brain function and use this as a molecule that links exercise to brain health. So here's what we propose to do. We're interested in answering the question, what are the behavioral effects of Lac-Phe on cognition using animal models? We're interested in the molecular identification of the targets of Lac-Phe in the brain that might be mediating its effect on learning and memory. And finally, we're interested in using synthetic chemistry to develop potential analogs of Lac-Phe that might be orally bioavailable and modulate learning and memory. With Tom Montine, my lab will be working on the first of these questions, and then with Stephen Bannock's lab, we'll be working on the second two of these questions. The idea of exercise as medicine has been around for a very long time. And we believe that with our recent discovery of Lac-Phe, we have a unique perspective on this problem and a potential opportunity to reduce it to practice and bring it to reality. And in the long term, what we hope is that Lac-Phe might offer an opportunity then to really therapeutically capture the molecules of exercise for brain health. So with that, I'd like to thank you for your time and attention. I'm happy to take any questions.
So great. So what's the biogenesis pathway of Lac-Phe, and is it only generated in the brain or elsewhere in our body too?
Yeah. So Lac-Phe is a chemical conjugate of lactate and phenylalanine. So it's intimately linked to lactate metabolism. Highest Lac-Phe levels are produced after sprint exercise when lactate levels are also increased the most. We've also identified the biosynthetic enzyme for Lac-Phe. It's a cytosolic enzyme called CNDP2. Knockouts of CNDP2 are the way that we produce animals that cannot produce Lac-Phe after exercise. And from a cell type expression, CNDP2 is actually expressed in innate immune cells like macrophages that are localized to diverse anatomical areas, as well as epithelial cells that are predominantly in the kidney and in the gut. So it seems to be potentially an endocrine circuit induced by exercise that regulates the brain, although we can't formally exclude possibility of, for example, glial neuron crosstalk in a paracrine manner.
You might have answered my question. So does aerobic exercise also create Lac-Phe?
The short answer is yes, but not as much as sprint exercise. In fact, for those of you who are exercise curious, the classical sprint assay that people use is something called a Wingate test. You can look it up on YouTube. It's commonly used in hockey teams and other sports that require high anaerobic power. What you basically do is a 30-second sprint on a bike. And it turns out that those conditions induce Lac-Phe the most. I've done it myself, it's true. But we also see after that, it's resistance training, and then after that, it's aerobic training. So we think that probably when this pathway physiologically kicks in the most is after hard, acute sprint exercise, although we see signatures of it across all different types of exercise.